So this data visualization system has been built in order to help biologists access radiation metadata associated with studies that have been collected by the NASA Gene Lab in order to better understand biology's response to environments related to spaceflight, such as low Earth orbit, the Moon, and Mars. So there are a few different interactive data visualizations that we've created in order to make these data accessible. And essentially they can be used to filter through the large number of studies that's in the GNAM repository in order to find some that have radiation values that you find interesting. We've also created a visualization system of the genes listed in the RAD gene atlas, a recent publication. So essentially to demonstrate how after identifying studies of interest using the radiation metadata, you can then go and look at those primary studies in more detail and look at their genetics, and then try and find genetic patterns and proteomic patterns associated with the response of biology to radiation. So at the beginning I have our first menu. So this kind of just divides the app into four main sections. You can either explore the primary data of the average dose rate, or you can explore the terrestrial radiation types. You can also explore different studies that have been performed in low Earth orbit, and also eventually a meta-analysis in which we've enabled the comparison of these radiation values associated with these space biology studies to be compared to robots and rovers that have been sent to the Moon and Mars. In order to help biologists understand some of the technical terms associated with this radiation biology data, we have created a glossary of terms at the beginning here to help people who are not experts in the field of radiation biology. So I'm just going to go along to the next dashboard. So this is the first way of kind of looking at the data. So we have these GLDS accessions. So these are Gene Lab Data System code names for studies in which scientists have investigated how biology has been responding to either the space-like environment or terrestrial analogs such as particle accelerators. You can see there's a lot of studies that have been pulled into this repository. So there's a lot of data that humans have collected, and some of them have had huge amounts of radiation given to them. So you can filter through that using this menu on the side. And so maybe there's a certain radiation range you're interested in, you can type it into the box there. In this particular view, you can also filter down using this slider. I personally like to define based on the clicking on the y-axis. And so you can then select a segment of studies, a collection of studies which have similar radiation characteristics. At the top here, you can see we have a summary of the number of organisms that are collected, the number of studies that you've got selected, the average and maximum radiation dosages that those samples got. Maybe you are a life science researcher, maybe you have a particular area that you're interested in. Some people are interested in human medicine, some people are maybe interested in mice, or maybe you're interested in microbiology and plants. So you can also filter through the study based on your favorite organism. So again, we're narrowing down the number of studies that we've got selected, and we're adding these filters at the top here. These filters can then be removed to enable rapid movement through this rather large data set. So let's have a look at the data in a slightly different way. So now we're looking at the metadata associated with the radiation source and the particle type. You can observe different aspects of the particle metadata, such as the particle energy inlet, the particle mass, or the particle charge. So some of the studies we have these data for, but a lot of the studies we don't, and this is because we don't have these type of data from LEO, from low Earth orbit, due to the type of detectors that are flown and the type of and then the nature of intergalactic cosmic rays. Let's go to the next way of looking at these data. So this dashboard is allowing us to now compare the spaceflight studies in particular. On the x-axis, we have the experiment duration in days, and on the y-axis, we have the absorbed radiation dose rate. One of the things that we've done is we've connected the radiation metadata associated with the Gene Lab accessions with values of radiation that were extracted from the Mars Curiosity rover when it was on transit to the red planet, and also from its range of different sensors. We've also been collecting metadata from other sources in the solar system. And one of that we've imported are from the Chang lunar lander from the dark side of the moon. And so we can now start to see some of the terrestrial studies in the context of these spaceflight robotic explorers. So we're just going to set a filter based on these lunar landers. Maybe bring that one up. And this then allows us to focus in on and find a smaller number of studies that we now think are kind of similar to the lunar surface. And so you can then filter through that and look at the sources. So now we've identified there's an interesting Drosophila study that might be worth a closer investigation. Once again, you can remove the filters from the top. On each of these dashboards, you can scroll down, and below we have like the metadata tables, which are easily accessible. And you can just click on the green button in the corner, and then download into your favorite kind of format, like tables. So essentially, this system is designed to search for GeneLab studies, GRDS accessions that you can then associate with different aspects of the spaceflight radiation environment. You can filter based on your favorite spaceflight vehicle, and you can explore other types of like radiation metadata and the diversity of the dosimeters that have been used in the spaceflight.